Hello everyone, today I'll be talking about RNA interference. A little bit of background about RNA interference. It was actually, scientists actually observed this in 1990 in plants and fungi, which was actually known as post-transcriptional gene expression. And in 2001, it was proven that short RNA interference could directly trigger RNA interference in mammalian cells. Fire and Mallow, who won a Nobel Prize in 2006, actually discovered RNA interference. And RNA interference is actually used to study the cellular function of gene expression. And it's used due to the fact that it is precise, efficient, and very stable. And there's several ways to actually induce RNA interference. And you can use synthetic molecules, RNA vectors, or do in vitro and dicing. Some applications about RNA interference. RNA interference identifies and assesses thousands of genes within the genome that potentially actually contributes to disease and phenotypes, and it provides an efficient means for blocking expression of a specific gene. And you can see here, I shared a little diagram showing the process of RNA interference. And next is the research article that I found. It basically focuses on foam cells and their role in atherosclerosis, which is a buildup of fat, cholesterol, and in and on the artery vessels. And it's basically caused by the ingestion of oxidized low-density protein. And the specific gene that they were trying to identify and was the RAB5, which is part of the GTPase. It's also known as RAS-related protein in the brain 5. That's the name of that. And what it does is a receptor mediator in endocytosis, and it's an early endocytosis. It helps with the fusion of endocytosis cells. And what they did is that they wanted to see the effect of RAB5 on macrophages cholesterol cells during the growth of macrophages when they are induced with OX-LDL, which is oxidized low density protein. And they wanted to see if that causes the formation of foam cells, which is basically foam cells are a type of macrophages that localize fatty deposit in the blood cell vessel cell walls actually. And what they found is that, if you can see here, they had three different sequences. They sequenced three different sequences here, and they had a control. And what they realized over time, that the presence of RAB5 was decreasing, and that actually short interference RAB5 75 showed that it downregulated RAB5, and it led to a 90% decrease of the level of RAB5 messenger RNA. And here it shows that by silencing the expression of RAB5 in the macrophage-derived cells, that after 24 hours, there was a downregulation of that specific gene that they were trying to delete or suppress, actually not delete, but suppress the gene. And they showed that there was no expression of that here. And here is the control, which has RAB5, So meaning it has the presence of it. And here, when they use the short interference Rab5 on these cells, you can see that there was zero observation of the red O staining. And finishing this article, there was a couple of questions that I added to the slides. And one of the question is, what are some disadvantages of using RNAi? And as of today, what is known about Rab5 and its involvement in facilitating the entry of OXLDL into macrophages and the formation of foam cells, which is still not known as today, because this article was written in 2017. And that was one of the questions the authors were trying to figure out. How can you get, also I was wanting to know, how can you get GFP template for the use of a negative control in interference RNA studies? And how is knockout of a gene on one side and knocking down the same gene on the other side affect the cell? So knockout is, if you've ever heard of CRISPR, that's what CRISPR does. It basically deletes a specific genes. It goes and it cut where the specific mutation is. However, RNA interference, it does not delete the gene. It suppresses the specific gene, so it's not expressed when it's actually not expressed. So those are the questions that I have.